Hi, good morning, everyone uh, who attended this seminar for today. For your information, my name is Silvia Berlanga Barros. I'm a professor at the School of Pharmaceutical Science, University of São Paulo, and I will join this seminar with you, presenting our speaker for today. This seminar is part of the activities of the course on topics in physiopathology and toxicology of the graduate program of the Department of Clinical and Toxicological Analysis of the School of Pharmaceutical Science of the University of São Paulo. The topic for today has experienced increasing importance in our days, considering the growth of biotechnological medicines and products used in different areas. The discussion about messenger RNA vaccines for COVID-19 vaccines brought the discussion of the safety of this technology. Regulatory toxicology has experienced a great improvement in the last century, but new approaches are necessary to deal with the safety evaluation of biotech products. This is the topic of our seminar today, who is toxicology and drug development in the area of biotechnology. To me, it's a great pleasure to introduce Professor Mary Ellen COSENZA. Besides being a great friend, Mary Ellen received her bachelor in biology and chemistry from the Queen's College, City University of New York, and his master and PhD in toxicology from the St. John's University, New York, and her master in regulatory science from the University of South California, LA. Mary Ellen, PhD, she's a member of, she's diplomated from the American Board of Toxicology, a member of ATS, ART, is a regulatory toxicology consultant with over 35 years of senior leadership experience in the biopharmaceutical industry in the United States, Europe, and emerging markets. During her 20 years tenure at Amgen, she was an executive director of regulatory affairs and safety and had several leadership roles, included head of US regulatory, head of emerging markets regulatory and safety and therapeutic area, lead for inflammation and early development. Previous to her role in regulatory affairs, she served as the senior director of toxicology at Amgen. Prior to join Amgen, she served as principal scientist in toxicology for the medical research division of American Cyanamide Company, who is now Pfizer. Mary Ellen is recognized as an expert in preclinical biological drug development. Is a founding member of BIO's BioSafe Preclinical Expert Group. She was also a member of an expert working group operating under the auspices of the International Conference on Harmonization, ICH, for ICH-M3R2. Mary Ellen is well published in the field of biological drug development, and she's a past president of the American College of Toxicology and is currently the treasurer of the International Union of Toxicology. She is also an adjunct professor at the University of South California. Before starting, I'd like to invite you all to, if you have questions, to upload your questions in the chat, in the YouTube chat, and she's gonna answer at the end of her talk. Thank you once more for attending the seminar, and thank you once more, Mary Ellen, for being with us so early, a little early in California at this time. Thank you so much. The podium is yours.
Thank you, Sylvia. It's my pleasure to be here today. I think we just need to have the slides put. Oh, there we go. Okay. So I'm going to skip the slide about me. You already heard all of that. And today I'm going to talk about just a couple of slides about where toxicology fits in drug development to set the stage. And then I'm going to mostly talk about biologics and toxicology. And if we have time, I'll talk for a few minutes about sort of the more newer, innovative molecules and biologics. So one of the reasons I love toxicology so much is that if you are in the field of drug development, pharmaceutical development, toxicology plays a role throughout the whole life cycle of drug development, from early research in terms of selecting early leads, all the way from filing the original IND or clinical trial application, all the way through all stages of drug development to the final marketing application. And then even after a drug is marketed, there are often toxicology issues that come up, either investigating things that show up in the clinic that we try to figure out why that happened if we didn't predict it, or perhaps new life cycle management, new indications, new populations such as pediatrics, where additional toxicology work may need to be done. This is sort of a typical view of how, particularly with small molecules, so many molecules are screened early on and then narrowed down to perhaps one drug many, many years later approved. And just to show that, toxicology plays a big role in narrowing this down. Now with biologics, we don't necessarily have this many molecules in the beginning, and I'll talk about that in a little bit. But again, just to sort of set the stage. Whether you're developing a small molecule, a large molecule, any vaccine, the goal of toxicology or safety assessment in drug development is to perform the necessary preclinical animal testing, and we also do non-animal testing as well, to support both clinical trials and then eventually the marketing. So some of the high-level goals for doing this are to identify drug-related toxicities. What are the potential target organs? Characterize the toxicity dose response. How steep is that dose response curve? Look to see whether the findings are reversible. Help identify clinical parameters to monitor as predictors of toxicity in humans. So what are the things in the early clinical trials the investigators should look for to know to maybe stop dosing humans, or to predict that certain findings might happen in humans based on what we saw in the animals. We look to help identify a clinical starting dose. So what's a clean, safe starting dose for the first in human studies? Evaluate toxicities that can't be directly tested in humans, such as those in the developing fetus, and develop an understanding of mechanisms of those toxicities we might observe. How do we know what studies need to be conducted to support which clinical trials? So there are a number of guidance documents around the world, and different countries have guidances specifically on first-in-human studies. The FDA has one for healthy volunteers. The EMA in Europe has one for all types of first-in-human studies. But probably one of the most defining guidances is the one that comes from ICH, the ones that come from ICH. And then the next slide, I have sort of a listing of all of the safety guidances. So in general, ICH-S, those guidances that are the different ICH S1, S2, et cetera, all of the different guidances that ICH has developed to help toxicologists know what studies to run and design those studies. There is one here in yellow you see, which is the overarching one that I actually got to work on the expert working group, and that's this one, ICH-M3, which is the guidance that tells us which studies to do when. It's mostly focused on small molecules, but there are some key principles that apply to biologics as well. It also mostly is about non-oncology indications. As a separate guidance, S9, which speaks specifically to those drugs that are being developed for advanced oncology. But it speaks to the timing of what studies you need to do when, and helps define those clinical starting doses that I spoke about. And I'm just going to go with 3 slides briefly on these principles. So for a phase 1 study, we usually do short-term toxicology, repeated dose toxicology, often up to 1 month. We do a genetic toxicology profile, and then safety pharmacology, which is really important because those studies look at cardiovascular, respiratory, and CNS toxicity, which are kind of the pivotal things that might happen in a first-in-human trial that could be life-threatening. So that's what we sort of do to support getting into the first clinical trials. And then as trials advance, we do longer and longer studies to match the clinical dosing regimen. And we start to look at things like fertility, teratology, the effects on the fetus, male fertility. And then for marketing, we do more detailed studies on looking at peri and postnatal, later development, and then carcinogenicity studies to support the eventual marketing. So that's sort of the general high-level paradigm. One last thing I'll talk about before I jump into biologics, and that's what I sort of call my Toxicology 101 slide. Very often people will say, oh, we ran a toxicology study and the study was clean. Well, clean toxicology studies are not necessarily a good thing for a toxicologist. There's no guarantee that that will happen in humans, but really an ideal toxicology study is one that has a toxic dose so that we know what the toxicities are that we want to try to avoid in humans, and then a clean dose that can help us define the starting doses. We also want to define that dose response curve, as I said. How steep is that slope? What are the different spectrum of findings in terms of all the different target organs? So I often say toxicologists are more concerned when there's no toxicity than when there's too much toxicity. And if you've studied the story of thalidomide, you know that the issue there was that those species, the studies weren't conducted in sensitive species, and so therefore a drug that looked clean actually turned out not to be clean in humans. So next I want to talk about the topic for today, which is biologics. What makes the study of toxicology and biologics different than sort of traditional small molecule pills, pharmaceuticals that have been developed in the past? So the thing that makes them most different is that these molecules are much more complex. They are biologics in general, and I'll talk separately about sort of more gene and cell therapies later, but biologics that we've been developing over the last 30 years are basically made by recombinant cell technology. They are biologics that are made of strings of amino acids. They have higher complex order, so it's not just even the chemical amino acid sequence, but the folding. They're manufactured in cells, and they often go under post-translational modification. So here you see sort of a picture of the structure of a simvastatin, which is a statin drug, and you see the chemical structure. And this can be made by organic chemistry in an organic chemistry lab, whereas this is a biologic, a growth factor, and you can see it's much more complicated. And it's not just the chain of amino acids, but it's the folding that makes this fit into the right place in the body to have its activity. Here's another picture just sort of showing, often you'll hear me use the term large molecules versus small molecules, and that's just because in general biologics are much larger. So here's sort of an typical antibody structure, and here's a small molecule, if you were to put it together, looking at the carbon and hydrogens, etc. In addition, one other reason we call them large and small molecules is because of the molecular size. And here's just a table sort of comparing a few of the traditional small molecules. And you can see that generally they don't go much larger than 500 or 600 molecular weight. But for large molecules, biologics, antibodies, you can see that they are up into the 147,000 for these antibodies molecular weight. So much, much larger molecules. How do we define what a biopharmaceutical or biologic is? Well, this is actually not as straightforward as you might think. Different countries actually have slightly different definitions. This is a definition that comes from ICH, so I'll use that one today since this is an international discussion. So ICH defines biologics or biopharmaceuticals as products that are derived from characterized cells, including bacteria, yeast, insect, plant, and mammalian cells. So very often the production of the biologic is done in cells that are either like CHO cells or E. coli cells. They're done by fermentation. Used to be done in like roller bottles, but now they're done in large vats. Kind of looks like brewing a beer. Includes proteins, peptides, and the derivatives. So if you have a antibody with a conjugate, a toxin attached, that's still a biologic because the main structure is the antibody. And some examples include cytokines, proteins, growth factors, fusion proteins where we've actually connected different pieces of molecules together, enzymes, receptors, hormones, and monoclonal antibodies. And monoclonal antibodies have probably become one of the biggest areas of the last few years. Another key attribute I'd like you to take away, if there's only a few things you remember from today's lecture, I'd like you to remember this term, that for biotechnology products, the product is the process. because again, it's not just that structure I showed you, the small molecule that you can make in organic chemistry, but it's all the folding, the stability of that molecule that makes it actually work or not work. And if it's folded wrong, it could actually be dangerous because your body could reject it as a foreign substance, and you can make antibodies against it, which I'll talk about in a little bit. So remember that the product is a process. It's not just the structure, it's all of those attributes. Some of the other characteristics I'd like to point out that differ between sort of biologics or large molecules and traditional drugs. One I just mentioned is the immunogenicity, that the body can, animals and humans can make antibodies against these molecules because they are so large, and if they're not made correctly and misfolded, if it's a recombinant human protein, but it's misfolded, it would be seen as foreign. The way we develop our toxicology studies is on a case-by-case basis, and I'll talk about that in a minute. We have a few issues that are really key, and I'm going to talk about this one a bit more in more detail, which is species specificity. Often these molecules are made to work in humans, and because they are proteins or biologics, they only work in humans, or they only work in higher order species, such as non-human primates. And so that can make the toxicology work more challenging. The other thing is they're not taken as pills. The route of administration is usually parenteral, IV, or sub-Q. They're not metabolized by the liver the way small molecules are. You don't have metabolites. They're generally broken down to proteolytic degradation, often by proteases in the blood, sometimes in the liver and some tissues, but that's how they're broken down. The half-life is longer, so sometimes antibodies can have half-lives of 3 weeks or several months, and so they're not dosed, every day or a couple times a day like your pill is, your headache medication, etc. And then we have a lot of issues with cross-reactivity. And so we have to make sure that the molecule is specific to the site we want it to work in. So, as I said, we often think about these as developing them from a toxicologist's perspective on a case-by-case basis. So it is true that in general, regulatory standards for biotechnology products have been more flexible because we have to design them based on both the characteristics that I talked about, the product is the product characteristics, the process is the product characteristics, as well as the pharmacology. So you can't really develop a biologic if you don't really fully understand the pharmacology, because most of the toxicology is going to be related to the pharmacology or to pharmacology we're not aware of. So sometimes we'll make a molecule to bind to a specific receptor, because we know that receptor is in a particular organ, and we do that, and it works in that organ, and it does the activity we want. But sometimes what happens is we see activity we didn't expect, and is that because the biologic is doing something else, or is that because the receptor is in tissues we didn't expect or we didn't know about? And that's in the past often been the case. So before ICH guidances were written, the concern for biologics was that they was more about some of the product attributes in terms of contaminants. It was felt that a lot of the early biologics were human replacement proteins. So the first one approved in the United States was recombinant human insulin. So the focus was on things that might carry over from those cell culture media. And the assumption was that the toxicity was related only to pharmacology. Some of this turned out not to be true. But if you were to look at the original package of toxicology studies that were done for recombinant human insulin, you'll see that it's mostly short-term studies, right? Only 1-month studies were done in terms of the length of trial. And all of these studies were really focused on potential carryover products from the cell culture, pyrogenicity, thinking there might be viruses or other cell host proteins that would be carried over. And so that's all that was done. And of course, insulin is used for, can be used for someone's life. And 1-month studies were the longest done. So that has clearly changed. Now our typical programs are go out to 6-month studies. That's what the guidance says. That for biologics, 6 months is usually sufficient to pick up all the toxicities. We look more now at safety pharmacology as well. We do reproductive tox studies. For antibodies, we look at tissue cross-reactivity studies or other studies that are done looking at receptor binding across panels. So you can see that for a typical replacement protein, this is sort of the standard toxicology program, as opposed to a small molecule program where there's longer-term studies done out to 9 months in your large animal species, and carcinogenicity studies, which we generally don't do for biologics. And these are the things that make these programs, longer and more expensive. So this is just kind of a typical sample program. So, as I mentioned, the program that I just discussed for recombinant human insulin was before ICH guidances were developed. And this is the guidance ICHS6, which has sort of become the classic guidance now for biologics. And so there are some concepts that are the same for biologics and drugs. Some of the things I talked about in the beginning of my lecture about what are the things we look for as toxicologists in our toxicology programs. So what are the known or anticipated risks? What toxicities would be monitorable in the clinic? Are they reversible? How do we manage these toxicities in the clinic? And what are the safe starting doses and dose-limiting toxicities? So these concepts are no different for small molecules versus biologics. We still want to look for these same attributes. But for biologics, there are some things that are specific that we have to think about differently than we do for small molecules. And that's the first one I'll talk about more in a minute, which is, are the toxicology species that we're studying our molecule in, is it pharmacologically relevant? And this can be a great challenge. Sometimes we look at animal models of disease because sometimes these molecules only work in diseased animals. There's no relevant receptor in a non-diseased animal. Immunogenicity, which I'll talk about more in a little bit, which is one of the key concerns or principles of doing toxicology studies for biologics. Things like gene toxicity testing. We don't do much of this with biologics unless there's a toxin attached to the molecule. Chronic tox testing, as I said, we generally only go out to 6 months. Tissue cross-reactivity studies are done mostly just with monoclonal antibodies, and we don't generally do carcinogenicity testing. And the study designs are often different because we try to make our toxicology study designs more similar to the clinical design, which can be very different for a molecule that is given IV or given only once every 3 weeks, etc. But we always try to keep the overall consistent idea of the 3 R's: reduction, refinement, and replacement. So we try to be very judicious with our use of animals in these studies. And then I'll just point out briefly that the original ICH guidance that I just mentioned was issued in 1997, and it talks about all this flexibility, which I'll talk a little bit more about in a minute. And then there was an addendum to this that was issued in 2012, and that helped clarify some key issues, some of which are the ones I'm going to talk about in a minute. So some of these issues were species selection, study design, immunogenicity, reproductive tox studies, trying to reduce the number of reproductive tox studies, and carcinogenicity. Different ways to look at carcinogenicity with biologics as opposed to doing standard 2-year studies in rats, which often are just technically impossible with a biologic. And that's often because these molecules don't work in rodents. Okay, so let's jump into sort of the key challenges. The first one being the selection of animal species. How do you decide what animal species to run your toxicology studies in for a biologic? Well, as I said, it has to be pharmacologically relevant. The guidance is very clear about that. So we often will look at the homology. If we're looking at a replacement protein, what's the amino acid sequence between species? How similar are they? Do you need to look at a disease model in order to show activity? If there is no relevant species, could you put the receptor into humans by making a transgenic animal? Or the other way around, can you make a species-specific version? So if the molecule doesn't work, the human molecule doesn't work in rodents, can you make a rodent version of the molecule that has the same activity? And then are there other species-specific findings we need to be concerned about? So as I said, the guidance is very clear. that safety evaluation programs should include the use of relevant species. And in the guidance, ICH, it says that's one in which the drug is pharmacologically active due to expression of the target receptor and epitope. And identifying a relevant species can be one of the most challenging things. And I can tell you, I've had experience years ago when I was head of tox at Amgen, where we did a lot of in vitro work showing that a particular antibody bound to the receptor in primates, very similarly to the way it bound to the receptor in humans. And we ran a tox study, and it was completely clean. And we knew that shouldn't be the case in normal, healthy animals. Well, it turned out that this molecule bound to the receptor in monkeys, similar to humans, but it actually didn't induce anything. It didn't go through the transduction, didn't turn the cell on. So binding itself or expression of the target is not sufficient. You actually have to show pharmacological activity. So this is just some of the different types of data that companies will do to try to figure this out. As I said, you can look at the sequence homology, you can look at receptor expression, you can look at in vitro binding, but as I said, that's often not enough. You can look at receptor ligand occupancy and kinetics, but you really have to look at the function. You need to know whether or not the way the molecule works is similar enough to be able to use the animal species. So sometimes we'll look at neutralization of the target and other types of cell assays. So, and of course, another way to do it is to actually look at pharmacology. Give the biologic, give your molecule to an animal, and does it do the same thing? And so here's an example where this is the keratinocyte growth factor, or Kepavance, also known as palifermin. It causes the oral mucosa that's been damaged from chemotherapy or radiation to grow back. And so you can see that here we actually gave it to animals, and in the control you don't see any growth, but with the growth factor you can see that in this animal these cells grow. And so we were able to show this in a number of species, and therefore the toxicology studies could be conducted both in rats and monkeys, because we actually were able to show in those animals the pharmacological activity. So that's one way of showing pharmacological relevance. As I mentioned, if your molecule doesn't work and you can't find a relevant species, you can always look at surrogates. So this would be an option if you have no cross-reactivity. Sometimes this is done in just for reproductive tox studies. So even if your molecule works in primates, as opposed to trying to run reproductive tox studies in primates, which are very expensive, take a lot of time, and really are only hazard identification, because you can't run a large number of pregnant animals, we'll develop a surrogate. This has its own challenges because surrogates take time. It's like developing a whole new molecule, so it may delay another project. So here, just sort of how we define what a surrogate is. It's a molecule that hits the same target. One example is that one of the developers of anti-TNF antibodies developed a rodent version and did the reproductive tox studies with that. You have to also it can also be used to just define pharmacology better. So sometimes it'll be just used for proof of concept in pharmacology models. And whether or not it's actually suitable to define toxicity for predicting what's going to happen in humans, because it is an animal version used in animals, is you have to think about whether or not that's actually going to help you predict the PK in humans, because it's a different molecule now, whether it will help you predict immunogenicity, et cetera. So it definitely has challenges. It's not the answer to every study or every project. One of the other things I mentioned is that with biologics, we often only run studies out to 6 months. And that's because when a review was done looking at studies done, chronic tox studies done with biologics, it showed that generally 1 to 3 months were able to support the clinical trials and generally picked up most toxicities. But for chronic tox studies, 6 months was the agreement in the addendum. So in S6R1, that's what was agreed to. And that picked up almost every toxicity seen with chronic tox studies, and studies of longer duration are generally not, don't really help define more toxicity, although there have been cases where the FDA or other agencies have asked for longer-term tox studies. The other issue that comes up is recovery periods, and this can be challenging because biologics have long half-lives, unlike a small molecule where you stop dosing the drug at the end of the study. Within a day or two, the drug is cleared, and so if you have a 2-week or 1-month recovery period, you really have a time where the animals have no exposure to the drug. But with a biologic, with an antibody that has a half-life of 21 days, you'd have to go for months in order to get to a point where there's really no exposure. So the guidance, the discussions that led to the wording in the guidance is that you don't necessarily have to get to full recovery. It's not really considered essential, but you can get to show a trend towards recovery. And also these recovery periods don't need to be assessed, either delayed toxicity or immunogenicity, that, you really just need to sort of show that the animals sort of trend back towards recovery. The other thing, as I mentioned, is we generally don't do carcinogenicity studies with biologics. So there's a lot of discussion on this. Obviously, there are some challenges, as I said, because a lot of molecules don't work in rodents. So you can't do a 2-year rat study with a molecule that doesn't actually have pharmacological activity. But you can do what's often called a written assessment. And so you kind of look at what you know about the biological activity, you look at all the literature, you think about what the patient population is, and what would be the potential to influence tumor life cycles. And if you think there is a concern, then you may need to do specific studies to look at the effects on that. But often a written assessment is considered sufficient. And the next big issue I want to talk for a few minutes about is immunogenicity. So as I mentioned a few times during my lecture so far today, is that because these are large molecules, proteins, antibodies, they will often stimulate the immune system. So the ICH S6 guidance says that most biotechnology-derived pharmaceuticals intended for humans are immunogenic in animals. Because it is the actual human sequence, or humanized or a fully human monoclonal antibody in a monkey, or even if it works in a rat, it's seen as a foreign molecule because it's not meant for monkeys, it's meant for humans. And so we know that these molecules can often cause immunogenicity in animals. And so we have to look at that. We have to measure those antibodies and look to see whether they have an effect on pharmacokinetics and pharmacodynamics. And that's what I'm going to talk about next for a couple minutes. So there are what we call clinically relevant antibodies. These are antibodies that have an actual effect on the either pharmacokinetics or activity of your biologic, your drug you're developing. So there are clearing antibodies, and the best way to describe that is to show a picture in a minute. These are antibodies that actually bind to your drug and make it clear away from the blood sooner. There are actually sustaining antibodies that have the opposite effect, that make the biologic stay around longer. There are antibodies that are neutralizing, that cause your biologic not to work anymore. And then are the most dangerous or concerning antibodies, and those are ones that cross-react with an endogenous protein. So you've made an antibody, you've made a drug, a replacement protein drug, like Epogen, recombinant human insulin, recombinant human growth factor. You give it to people, they make an antibody against your drug, and now that antibody binds to their own endogenous version, and that can be, very devastating. And the last thing I'll mention is that you can also have these complexes, the drug now that with the antibody that's bound to it, and those complexes themselves can cause toxicity by either depositing in tissues like the kidneys, etc. So here's a couple pictures I think that help describe some of these antibodies I talked about. So here's a classic clearing antibody. So you give the drug to an animal, and here's your PK profile. You can see that you can measure levels out to almost 21 days. That's sort of typical of maybe an antibody or a large replacement protein. And you give the dose maybe once every 3 weeks or once a week. And when you give to the third dose, particularly if it's once a week, now your body or the animal's body has generated an immune response, an antibody to that drug, and you can see now the PK has changed. So it's made it disappear, clear faster, and you can see that it's gone in a couple of days. So that's called a clearing antibody. Sort of the opposite of that is a sustaining antibody. This is an antibody maybe to a smaller biologic where it actually makes it hang around longer. So you can see at the end of a 1-month study here, we looked at the PK in animals, and we were able to break them down into sort of 2 groups. Those animals that had no antibody made against the drug, and so that was their PK profile in terms of hours. But for those animals that did have an antibody that was sustaining, you can see that the drug stayed around longer, and actually was much more variable because different animals have different amounts of antibody that they make. Here's another classic example of a clearing antibody where you can see on day 1, all these animals, their individual profile is very similar, looking in terms of days, how long this molecule stays around. But when we look on day 15, you can now actually look, by looking at the PK, you can actually tell which animals have an antibody. These animals have an antibody that's clearing, and these animals don't. And as I mentioned, the one that's most concerning is when an antibody is made to a drug, but then that cross-reacts to an endogenous protein. And so here's an example where we're looking at a biomarker. Perhaps this is red blood cells or platelets, whatever it is we're trying to make a growth factor to do. You give the drug to the animals or to people, and you see the rise in the biomarker you're trying to measure. You give it again, you get another good rise. And then you see that biomarker start to go down. You give the drug again, and not only do you only get a tiny rise, but you see it actually crashes because now that patient or that animal has made an antibody against the drug and their own endogenous molecules, such that now they become either completely anemic or they have almost no platelets. So this is actual data from a clinical trial. So this is really what we try to avoid. And that's why you don't see as many replacement proteins now. The last one I'll mention is just this idea of a neutralizing antibody. And sort of what makes this different is that you can have antibodies made to your drug that change the PK but don't change the pharmacology. But a neutralizing antibody is one that actually changes the pharmacology. So here you see you have your drug. Your drug here is in green. It has a non-receptor binding part of the molecule, and then this is your functional part of your molecule. And the way your molecule works is to bind to a cell receptor. It turns the cell on, and it has its activity. Here you can see the person or the animal makes an antibody to the drug, but it binds to the sort of non-functional part. And so the drug still binds to the receptor and the cell works. And so that's a non-neutralizing antibody. In this case, your antibody that the person or the animal makes to the drug binds to the functional part of the drug, and now it can't bind to the cell, and so you have no cell response, and that's called a neutralizing antibody. And often we'll define these by creating cell assays that function this way so we can see whether antibodies are actually neutralizing or not. So in the next few minutes, I'd like to talk just briefly about some of the newer biologics. What I talked about was mostly sort of traditional biologics, replacement proteins, antibodies, fusion molecules. Today, we have many, many more types of biologics. I'll just mention a few of them. We have autologous products where a patient's own cells are taken out and manipulated in some way. We have cell therapies, immunotherapies, CAR T cells, antibodies that are combined with other molecules, gene therapies, and oncolytic viruses. And I don't have time to go over all of these. I'll just mention sort of a few that have become more common recently. So autologous products are those where a patient's own cells are removed from their body and then manipulated in some way and then put back. And so these are some of the typical ones. Cytotoxic T cells or dendritic cells are commonly used to attack tumor antigens. Tumor vaccines are often made this way as well. I know monoclonal antibodies are the more traditional ones I talked about before, but I just want to mention there are some that are sort of seen as more innovative now because they work in conjunction with some of these other molecules I'm talking about, and that's ones that either make a cancer more visible to the immune system or block growth factors or block blood vessels from growing. And so here's just a cartoon of some of these specific types of antibodies that are used to have these more specific effects. And conjugates, I mentioned, that's sort of become much more common where an antibody is bound to a toxic conjugate. The antibody binds to a receptor on a tumor cell, and then it actually kills those tumor cells. Or that's also can be done with radiation, where then radiation is used to kill the tumor cells. And this has led to sort of also making new structures of antibodies. So as opposed to a whole antibody, maybe we make an antibody that has multiple arms or take pieces of antibodies. You may have heard the term bispecifics. So this is where either you have an antibody where the different arms now bind to different targets or pieces of antibodies. And the first one that was approved in the United States was one that's called Blincyto. And I can show you a little cartoon of this. It's called a BiTE, and blinatumomab is the actual marketed product. And what here, it's not the full antibody, but it's just this little piece of two different antibodies are brought together. And that's what's called the bispecific or BiTE. And what this does when it gets in the body is it brings two different cells together. So the cancer cell here, which is an ALL blast cell with a cytotoxic T cell, activates that, brings those together, and causes apoptosis. It also causes proliferation of these T cells that now recognize this cancer cell. And so it's this is, as I said, the first bispecific that was approved in the United States. T cell therapies are therapies where T cells are taken from a patient. They're often exposed to a virus that has a gene that we want to put into the T cell, and those T cells are then expanded and reintroduced back into the body. Here's just showing a little more how this can be done by actually using sort of a gene therapy on the cells to put the message that you want those cells to express. Dendritic cells are antigen-presenting cells, and so there are different things that can be done with this, but one way that these are developed is T cells are taken from a patient. They're actually stimulated to form dendritic cells. They're exposed to antigens, specific tumor antigens, and then reintroduced back to the patient with the idea that then these T cells or these dendritic cells will go and attack the patient's tumor. Probably the one you've heard a lot about recently has been CAR T cells. So these are basically where a clone of a chimeric antigen receptor is put into a T cell. This allows this cell to recognize and bind those tumor cells. There's been very encouraging work in different leukemias and lymphomas with this. There's more work being done now on solid tumors. That's been a little bit more of a challenge. This is just a cartoon showing what a CAR T cell looks like, where you actually clone in this receptor, which then binds to the tumor cell. And I'll talk a little bit in a minute about what we do from a toxicology perspective with these types of molecules. But just to show that this has become much more almost mainstream now, from the early studies where now we have several CAR T cells approved in the United States. And again, this is really where the function is in this receptor. That's this clone. This is cloned into the cell, and then the cell expresses this receptor, which then binds to the tumor. So from a toxicology perspective, what we're really concerned about, because this is a patient's own cells, generally these are autologous, is we're concerned about what is that binding section that we put into those cells now, what does that bind to? And so we'll look to see, target profiling to see where those targets might be. We look at what's sort of called on target but off tumor binding. And the other thing we're often concerned about with these products is cytokine release. And that's been one of the biggest side effects seen in the clinic. And so we'll do all kinds of different studies to look to see whether cytokine release is a concern. They'll also look at tumor-bearing animals to see what the activity is there, and you can sometimes collect toxicology parameters in those tumor cells. People have at times looked at making homologous CAR T cells in animal models, but I would say in general, the agencies have been discouraging of that type of work. Hasn't really, I think, been helpful. But the other thing that's going on is we're making different versions of these CAR T cells. They're not always autologous anymore. There are a lot of allogenic CAR T cells where sort of off the shelf. We make them from normal healthy volunteers and then have them there ready to give to patients as needed. Also adding sort of novel suicide genes. So if the cells get make too many, how you can turn them off by giving the patients a drug. And the last thing I'll talk about just really briefly is gene therapies. This is another very growing area. There aren't very many gene therapies approved yet, but there are a few. And this is basically, we define gene therapy as medical intervention based on the modification of genetic material in living cells. And they can be developed to be therapeutic, prophylactic, or marking cells. But therapeutic is probably the one we're most interested in. And so what generally is done here is that we take a viral vector and we take out some of the genes that encode for the making more cells or making the viral envelope, and we put in our favorite gene instead. So we take out these that make more virus and we put in what we want the cells to make, and we leave this part in. We put this virus, this gene therapy into the cells, and now this will make this what we call a transgene product. So if a patient doesn't make a particular protein, that's what their disease is. They can't make a particular protein. They're missing the genes to do that. We put the gene into the virus gets into the cells, and it integrates into the nucleus, and now they can actually make that protein. And that's really the goal behind gene therapy. And one example that has had some success in recent years has been with what's called SCID, or severe combined immune deficiency. You may have heard the term bubble baby. There was a movie many, many years ago with John Travolta called The Boy in the Plastic Bubble. These are children that are born without a fully functioning immune system. And it can be caused by a number of different missing genes, but one is the missing ADA gene. And so in these children, they take out their bone marrow, they treat it with the gene therapy, the virus, to put this gene in to give them back the ability to make this protein. These cells are put back into the child, and there has been some success, as I said. This is just one example of a little girl that was treated at UCLA who has now been cured of bubble baby disease. What do we do in terms of toxicology for these kinds of really novel molecules? Well, we have to really think about what is the proof of concept. What can we find out from running studies in animals that would help us predict what we would see in humans? And so it's can be very, very challenging to do this. And so these are cases where we often want to talk to the FDA or regulatory agencies very early. So we'll look at, where the product goes, what we would expect making more of that gene would do. What if the virus goes the wrong place for gene therapy? What if the cells can't get turned off with a CAR T cell? But we have to always think about the 3 Rs. Sometimes people will want to do animal studies just because they can, but that's not necessarily a good use of animals if it doesn't help us. And I just mentioned here, we still have to follow the GLPs. So I'll just mention lastly, in the US, because these are very complicated products, the FDA has developed a sort of a new program called Interact, which allows you to have very early meetings with the agency to discuss these advanced products. And it's really only specifically for cell and gene therapies and other types of advanced products. And they have what's called the CAT team, which also is available to just talk about new technologies, even if you don't have a molecule yet, but you want to talk about a new technology. And they've also have an incentive program specifically for these, and that's called RMAT, Regenerative Medicine Advanced Therapy. There have been a number of products for that have been developed using this now. It's specifically for serious or life-threatening diseases. There has to be an unmet need, and the FDA can give you this designation. And there have been some products approved here in these categories I just talked about. So these are the first two here are gene therapies, and these are CAR T cells here. And in summary, toxicology data is critical for helping characterize the overall safety profile of pharmaceutical candidates. The contribution of toxicology spans the whole life cycle of a pharmaceutical drug development. So I think that's really important and one of the great reasons to work in the field of toxicology in drug development. There's a lot of different regulations, different health authorities, but as I mentioned, ICH can be really helpful. And you need to think about things like the drug's properties, as well as your target population, clinical trials. As I said, health authorities are generally more flexible for agents that treat life-threatening indications or for those without available effective therapies. And the last thing I'll just mention again is that to develop a biologic, you really have to have good knowledge of the pharmacology and molecular aspects. And I'll stop now for some questions. And here's my email address. I have some references here at the end that people can look at. But feel free to also follow up with me if you have questions by email. So I will turn it over to see if there's any questions, Sylvia.
So, Mary Ellen, thank you very much. So you did a stupendous, awesome job covering in this short timing such a complicated. But I hope that everyone has a lot of doubts. But anyway, so we have one question I'd like to ask about the long-term treatment using in vitro studies. Oh my God.
Yeah. So in vitro studies can be really helpful to define mechanism of action, look at activity, at a cellular level, but they're not going to give you sort of the long-term effects on organ systems. And of course, as you know, in vitro, cells only last a short period of time. So it's not really possible to study long-term treatment with in vitro systems. And that's why we still use so many animals for our studies.
I'll make a very brief comment on that. If you talk to people from Emulate, who is probably one of the first companies that develop in vitro methods, they will say that probably they can develop methods that can, for example, be like 15 days, 30 days, but for sure the chronic effects are really difficult to address.
Yeah, I think we should be looking more and more at finding more in vitro assays, but again, they really help more with mechanism than necessarily with chronic exposure in a whole body, right? And that's the challenge, is we still haven't really developed good ways to sort of mimic the whole system biology in vitro, even using organs on a chip and more complex in vitro assays, not just cells. It's still difficult to sort of balance that whole, in the body we have all kinds of different hormones. You have your lymphatic system, your hematological system, and it's difficult to mimic all those in a test tube, so to speak. So I see there's a question on homologous proteins. So homologous proteins are really helpful also, but again, they can be different. So actually, as this question says, it can have similar activity, but it could be there can still be differences in the way they work. But they have been helpful, particularly with, I would say, mostly with reproductive tox, where we are then able to test the homologous protein in rodents, because the reason we like to use rodents for our reproductive tox studies, for our teratology studies, is that a pregnant rat can have 15 offspring, right? So by dosing one animal, you can look the effect in a large number of fetuses, which isn't the case either in humans or in primates or many other species. So we'll often make surrogates specifically for reproductive tox studies that we can use in rodents. You can dose 10 rats, and you can have maybe up to between 100 and 150 fetuses to evaluate. And so that gives you a really good sense of a safety profile. Where if you do the actual human molecule in monkeys, in pregnant monkeys, they have a long gestation period like humans. It's one monkey, one baby. And so you can't really dose enough monkeys to get that same sense of safety. You might be able to do what we call hazard identification, but that doesn't really, give you a good safety profile.
So
I see there's also a question about CAR T cells. So yeah, CAR T cells can also be used post-plant transplantation. They're being used in all kinds of different ways now. The challenge, I guess, to focus on the toxicology part, is to try to define what the potential toxicities are, whether the side effects are of a CAR T cell, because these are very vulnerable patients, right? Particularly if they are being treated with other immunotherapies, or if they're post-plant transplantation, they're very vulnerable. And so you want to make sure that the molecules are very safe. And so we often will look at both the side effects, cytokine release being probably the biggest one, but there's also been neurotoxicity, where sometimes for reasons that's not fully always understood, that's often more about the patient than the cells. Patients have had neurotoxicity findings, seizures, etc. But also you want to make sure that CAR T, the binding section that now your cell is binding to, that it only binds to the cancer cells, that it doesn't bind to normal tissues, as I said before, just to reiterate that term, on target but off tumor, right? So is this a receptor that might exist in other tissues in the body, not just the cancer cells? Artificial intelligence. Well, I would say it's probably not been a lot of activity with artificial intelligence. I mean, there is, I did actually work years ago with some in what we call in silico modeling. So for small molecules, there has been some, there's programs like MCASE, TopCat, Derek, that do look at the structures of small molecules, and that can predict some very minor attributes. Not that they're minor importance, but there are only a few, such as genotoxicity. So they have gotten pretty good at predicting whether a molecule might be Ames positive or not, but there hasn't been a lot of great activity or great predictability of using these computer modeling for other types of toxicities. Maybe cytotoxicity occasionally, but that's not really the things that those are things that we can do in vitro pretty easily as well. They haven't really been good at replacing animals.
Yeah, that's true. It's a long way to. So I think that we are at 11 o'clock, and you did a great job, Mary Ellen. What I'm going to say to all of you who attended, and mainly for the students, if you have any doubt or if you need more information and you didn't got the email from Dr. Mary Ellen, please send me that I can put you in contact. And also for the students, the video will be, I will send, I will ask Professor Marielle for the slides to send to the students that attended, and we can know who are the students because of the frequency of to the course. And after 15 days, I will ask to the communication people to open for the YouTube, because this is a so great presentation that I think that should be spread all over the world. That's true, Marielle. And I know, it's not easy because it's so much information, and so well designed in your slides that it will help a lot of people who really want to understand a little bit about this big challenge, who is the new challenge for biotechnology. It seems that easy to do the biotechnology products nowadays, but it's so easy to evaluate its safety.
Yeah.
That's my conclusion. So we don't have more questions in the chat. So I want to thank you very much, and I hope that we can meet in briefly in other situations for sure.
It was my pleasure. It was really a
Thank
pleasure.
you.
And I hope to visit in person one day.
Oh, I hope so. Thank you so much, Maria Ellen. Have a great day. Thank you.
Okay.
Bye,
You too.
dear.
Bye bye.
See you.